So the previous segment of this lecture ended with these questions, so let's go through them. A plant has wrinkled seeds. What is its phenotype? Well, this one's easy. The wrinkled seeds is how the thing looks, so the phenotype is just wrinkled. Okay, so let's look at the same plant. It has wrinkled seeds. What's its genotype? Now remember, in this case, wrinkled is recessive, therefore the genotype has to be the recessive allele, and by our convention, we use the lowercase letters when we use letters to represent the recessive alleles. Now that doesn't mean that every time we use a lowercase letter it's recessive. Remember, recessiveness is rare, but in this case there is dominance recessiveness relationship, and therefore the wrinkled seeds then would have genotype little r, not WW, remember. Okay. Now a heterozygote has what phenotype and genotype? Well, let's start with the heterozygote is telling you actually what the genotype is. The heterozygote has different letters, and again we're talking about the same trait. So in this case, the heterozygote would have genotype big R little r, and big R little r, since the big R is dominant and big R makes round, this would be then a round. Okay, and again, like I mentioned before, we're not saying RW because that would indicate that there are two different genes. That's our convention, the professional convention. Even though you'll see in some textbooks that appears to be the textbook author's choice to try to simplify it and make it easier for you. But actually it makes it more difficult, because then when you actually get into the profession it becomes somewhat confusing, because if you were to write this, people would interpret this as being only haploid and having two different genes. Okay, now what about yellow cotyledons, the yellow shape, or sorry, the yellow color of the seed? A plant has yellow cotyledons. What's its genotype? Now, in this case, remember, yellow cross green gave us yellow, which tells us that the yellow is dominant. In that case, then we don't know. We're not sure exactly what the genotype is, but we can at least say what it could be, because it could be homozygous dominant or it could be heterozygous. So now notice we can't use R here. We have to use a different letter because we're talking about a different gene. So the different symbols represent different genes. In this case, the symbol for the gene seed color is Y, and it comes in two alleles, big Y, little y. Now we can write that as any way we want. We could make it A's or B's or whatever. It doesn't really matter, as long as we define how we're using it, and as long as we follow the professional convention of one symbol for the gene and different forms of that symbol for the different alleles. Okay, so this then would be big Y would give us yellow cotyledons, but also big Y little y would do the same thing because yellow is dominant. All right, so far so good. So that gives us a description of what's happening there, an explanation of what's happening. But the other part that he had to deal with was this. Why is it not exactly 3 to 1? Why is it only close to 3 to 1? Well, let me play a game with you for just a moment. Suppose we go to Vegas and somebody's on the street and they say, hey, look, I want to play a really simple game with you. I'm going to flip a coin 4 times. Okay? Now, you can bet on whether or not you're going to get 2 heads and 2 tails, or on whether you're going to get something different. Okay? Now, which would you bet? What would you take? Okay, the guy's going to flip four times, and you can either bet 2 heads, 2 tails, or you can bet something other than 2 heads, 2 tails. All right, so we know that the expected result is 2 heads, 2 tails. So some of us might be inclined to take that bet. Say, okay, yeah, I'll say it's 2 heads, 2 tails. If you were to do that, that would be the mistaken bet. It's more likely that you don't come up with the expected result, even though that's the expected result. If the coin is fair, you should get half heads, half tails on 4. That means 2 heads, 2 tails. But that's, even though the expected event, it's not the most likely event. Okay, now, I want to go through that carefully with you here shortly, because this type of problem is precisely why it is we're not getting 3 to 1, but there's a problem here. And that is that the human brain does not think well intuitively in terms of this type of problem. And I know that for a fact. And the evidence that I will present is Las Vegas. Las Vegas is in one of the harshest deserts in the world. It's probably the harshest desert in North America. And yet there's this big, giant, thriving city there. Why? Because people from all over the country fly to Las Vegas. These are people who don't like to pay taxes. They don't like to pay for streets or schools or anything like this. But they leave their money there. They leave their money there not because they are trying to be altruistic and give money away. They leave their money there because they don't understand this kind of problem. Okay, so what are we talking about here? We're talking about this concept of randomness. A random problem, a random event, is any event the outcome of which cannot be predicted with certainty. Okay, like a coin flip. Flip a coin, that's a random event. Okay, so that's the definition of random. That's the technical definition of random. It doesn't necessarily mean surprising. It doesn't necessarily mean anything along those lines. It simply means that you can't predict the outcome of the event. Okay? Now, if you have a sequence of random events, we call that stochastic. You'll hear people talk about stochastic and random as if they were the same term, and they're not really. Random just means an event that can't be predicted. Stochastic is a series of such random events. Okay, let's go back to our problem. We're in Las Vegas, and this guy comes up and wants to flip a coin in front of us. Now, we can calculate what the probability is of having a particular outcome. Now, probability, there are a couple of different ways of looking at probability, which are not quite exactly the same. But the way I'm going to talk about here is what we call the classical definition of probability. The classical definition is this. If the coin is fair, and I flip it over and over and over again, then by definition, it being a fair coin, it has an equal probability of coming up heads. If I were to flip it an indefinite number of times, I get approximately 50-50 heads, tails. And in fact, technically speaking, if I were to flip it an infinite number of times, that frequency would converge to exactly half heads and half tails. Okay, so that would be the definition of a probability of a fair coin, which means the definition of probability of 50-50 chance. Okay, so let's take a look at this. If we have a fair coin, we flip a coin, we know we have a half chance it's going to come up heads, half chance it's going to come up tails, right? Now, can you calculate the probability that in that coin flip you would get, say, 4 heads and 0 tails? And the answer is yes. It's a very straightforward sort of thing. You use this thing called the binomial theorem. You'll learn that later. But if you do that, here's the probability of 4 heads and 0 tails is 0.0625, which means 6.25% of the time that you flip this, you should get 4 heads, 4 tails, or another way of saying it is, if you were to flip the coin 100 times, you would expect to get 4 heads and 0 tails about 6 and a little bit more times, right? So that's the concept. The probability of getting 3 heads and 1 tail is a quarter. Probability of getting 1 head and 3 tails is the same. It's a quarter. You see the symmetry. So the probability of getting 0 heads and 4 tails is the same as the probability of getting 4 heads and 0 tails. And that's because the coin's fair. But now look at this. Here is the expected outcome. This is what we say is the expected outcome, 2 heads, 2 tails. The probability of that is only about 1/3. It's a little bit tiny bit bigger than 1/3, about 37.5% of the time. So what that says is this: if you take that bet where you say, yeah, I'm going to bet on the expected frequency, I'm going to bet that you get 2 heads, 2 tails, you're going to lose 2/3 of the time. So this is the point. Why is it that in Las Vegas there are people who live there and build gigantic casinos and other people who go there and lose their money? The difference is mathematicians. The people who own the casinos hire mathematicians to do these kinds of calculations, much more complicated ones actually, for them. And they use that information to actually rip off their customers. All right, so this is the kind of thing, though, we have to think about. We have to see what these probabilities actually are, because that's what's going on with this deviation from 3 to 1. Why is there a deviation from 3 to 1? It has to do with randomness. The choice, the event that occurs that determines what the genotype of the offspring is going to be, is random. We're going to come back to that concept in a little bit more detail in that context in just a moment. Now that we've seen Mendel's explanation, what he's got to do next is to demonstrate that the explanation actually gives him the results that we expect. Now, if you remember, his explanation really only had 4 statements. The genetic material is particulate and not fluid. There is 2 particles for each trait, 1 particle from each parent, and one of those particles dominates the other when they occur at the same time in the heterozygote. From that, he says, we can explain why it is we got a 3-to-1 ratio in the F2, but not exactly 3-to-1 ratio in the F2. Okay, so what we want to do is go through that and look at that carefully. Here's what we're saying. In the first breeding, remember he started out with a wrinkled true breeding variety and a round true breeding variety. And when he does that, when he mixes those together and he breeds those together, all the offspring were round. Okay, now if his explanation is correct, then here's what's happening. The wrinkled individual must be little r, and the round individual must be big R. Now how does he know that the round individual is big R? Because one of the problems that we face is the fact that big R little r also gives you round, just as round as big R. But remember, go back to what he did before he started his experiments. When he started the experiments, he said, okay, well, look, I'm going to take these from the seed producer, and I'm going to test them over generations to make sure that they are in fact true breeding. So he grew hundreds of these plants, he forced them to self-fertilize, and then he looked at the offspring and made sure that the offspring always looked just like the parent. And that was the case here. So having done that over a number of generations, he knows that these must be big R, because if they weren't, if any of them were big R, little r, if any one of them even were big R, little r, he would almost certainly have gotten some that were a mix of round and wrinkled, and he never saw wrinkled at all in that lineage. So he takes these guys and he knows that they're big R, and he knows that these guys are little r because they're wrinkled, if his hypothesis is true. Okay, now the next statement is that in any given individual, there's two particles determining the trait, one from each parent. Okay, that means this parent is going to give one of these little r's, not both, to the offspring, and this parent is going to give one of the big r's, and that's what we're representing here. This is one of the big r's coming from this parent. It could be either the left or the right one. Same thing here. But in the offspring, one parent gave the offspring a big R, the other one a little r, and so the genotype of this individual is big R, little r. Now, in this case, then the fourth statement is that one of these dominates, and the one that's dominant is the one that you see in this heterozygote. Therefore, this individual who's heterozygous is round, therefore round is dominant. Okay, so far so good. Everything that he has said here is just diagrammatically represented in this picture. Those four statements are particles, one from each parent, two per trait, and one dominates, can explain what's happening from the parental generation, the F1. Now what he's got to do is explain from the F1 what happens in the F2. Okay, so what he's going to do now, remember, is he's going to take these heterozygous individuals, if his hypothesis is correct, and breed them together. Okay, so he does that. And when he does that, now we have to look at this in the same sort of way. Here is the female portion of the plant. And the female portion of the plant, remember, they're self-fertilizing, so they have the same genotype. The male and female portions have the same genotype. The female must be big R, little r, because she came from this breeding. Okay, and the same thing with the male portion of the plant. He must be big R, little r. Now, Mendel says each individual gets only one from each parent. Now, this parent, the mother tissue, can only give either a big R or a little r, and the probability that you get big R, little r is half. So we represent that by drawing this out, saying, okay, this is going to be a big R on this side and a little r on this side. You've probably done this before. This is not how Mendel did it. Later I'll show you exactly how Mendel did it, which is a much more powerful way to approach it. But anyway, we have then a big R here and a little r here. Now, these don't represent letters. They don't even really represent alleles. What they represent are gametes, meaning eggs in this case, because this is a female. So this is an egg. You should see in your mind that this is an egg that is carrying a big R, and this is an egg that's carrying a little r. So this egg can be fertilized by sperm cells from the father tissue, the male tissue of the plant. And again, same sort of thing. The male is only going to give one of these two letters, either big R or little r, to its offspring. So here are sperm that are carrying big R, and here are sperm that are carrying little r. And again, half of the sperm are carrying big R and half the sperm are carrying little r. All right, if that's true, then what this matrix represents are all possible matings between the two of these things. So in this case, here you have a sperm carrying a big R that fertilizes an egg carrying a big R, and so the offspring must be big R, which would make its phenotype round. This box here would be a big R sperm fertilizing a little r egg, which makes a round individual because it's heterozygous. Same thing here. In this case, though, however, the sperm is carrying a little r, and the egg is carrying the big R. So that's what's going on in this box. Again, you get a round heterozygote. And in this case, you get a little r sperm fertilizing a little r egg, and that gives you then the wrinkled individual here. All right, so we look at this, and we get 1, 2, 3 round to 1 wrinkled, and that's what he saw. He saw a 3-to-1 ratio, except for one problem. He didn't see exactly 3-to-1 ratio. It was always close to, but not exactly 3-to-1. Why was it not exactly 3-to-1? For precisely the same reason that when you flip a fair coin 4 times, you don't get exactly 2 heads and 2 tails every single time. Randomness. The choice in this case of gamete that it gets fertilized, the choice of the egg that gets fertilized, and the choice of the sperm that fertilizes it is randomly determined. There's a 50-50 chance that a big R egg for any given individual is going to be fertilized or a little r egg. Same thing with the sperm. So that means this, it's like we come up here and we flip a coin. We say, okay, well, if I get heads, I'm going to choose an egg with a big R, or I'm going to choose, if I get a tail, I'll choose an egg with a little r. Same thing here, heads give me a sperm with a big R, tails gives me a sperm with a little r. And that's the randomness, that's where it comes in. And so it's possible, just like we saw before, if you flip a coin 4 times, you could get 4 heads. It happens 6.25% of the time. In this case, then, if you have 100 offspring, it's possible you flip a coin, you always get a big R. It's possible. It's unlikely, but it's possible. And if that happens, then everything is going to be round. There won't be any wrinkled at all. That's by luck, by chance, however you want to say it. But the point is, that's what's deviating us from this 3-to-1 ratio. So Mendel knew that, and he understood that. And in fact, he dealt with this in a much, much more sophisticated way. This particular technique right here is called a Punnett square. And it works really, really well when we're first teaching this to students. And so therefore, that's probably why you've seen it before. Mendel was a mathematician. He used a much more sophisticated technique that allowed him to solve much more complex problems. And I'm going to show you that technique later. By the end of the course, you'll never use Punnett squares again. You'll always use the technique Mendel used, because it's actually much, much faster and much more powerful. But for right now, this is a good way to see what's happening. So what have we done here? What we've done is simply said this. We have now looked at the hypothesis in detail to see exactly what it's predicting, and it predicts exactly what he saw. A 3-to-1 ratio-ish. Almost 3-to-1, but not exactly 3-to-1. It's 3-to-1 because this pattern gives us a 3-to-1 ratio, but randomness pushes us slightly away from it. Okay, now what's the next step? Well, the next step is to test it. But before we test it, I just want to show you another example from a human example that will help us sort of hammer down exactly what's happening here. This human example is corneal astigmatism. Now, astigmatism is a typical sort of thing that occurs in human eyes. We all have astigmatism of some sort. And it, in essence, and I'm going to say this, ophthalmologists are going to be upset with me, but basically what it basically does is it makes part of your eye farsighted and part of your eye nearsighted. The most common form of the inherited form of it, you can get astigmatism for all sorts of reasons, a smack to the eye, or sometimes you're just born with a misshapen eye. But the most common form that's inherited, that's genetic, is called corneal astigmatism. And that's when the cornea, which is this structure that sits on the outside of the eye, bulges out slightly. Now, the eye anatomy basically works like this. The cornea is the outer portion of the part that lets light through. Beneath that is a water-filled chamber called the aqueous chamber. And then you have a lens here, and then you got this vitreous chamber here, and then the retina's back here. So light comes flying in through here, goes to the pupil, which is the opening right here, and goes back to the retina and gets transduced. That's how you actually see. The majority of the focusing actually occurs here at the cornea. The lens doesn't do much. The lens just kind of fine-tunes the actual focusing. Some people have a bulge in this cornea. Now, the cornea itself has to have the right parabolic shape, otherwise it won't focus the light properly. But some people have a bulge in it, and it's genetic, and that causes, again, the light to sometimes focus back behind the retina when other parts are focused on the retina, and some parts focus ahead of the retina, and so forth. And so that's why you end up with this wedge. People who have this condition have a wedge in their visual field, which is out of focus when the rest of it is in focus. It can be corrected, but this is, again, the most common form of genetic inheritance. Okay, so let's take a look at this. In this particular case, individuals with genotype spade. Now, I'm using this notation to get away from the concept that big A and little a mean whatever they mean, or big this and little that, because we generally don't use that in the profession. We use different symbols than just letters. But in this case, I'm going to say that one allele is spade and the other allele is club. All right, so individuals who have genotype spade have normal vision. Okay, individuals with genotype club have astigmatism. The heterozygous individuals, spade club, also have astigmatism. Therefore, which of these alleles is dominant, the spade or the club? Okay, now go back to the definition of dominance. Dominance is if the condition is expressed in the heterozygote, then that's the dominant condition. Right? So here's the heterozygote, spade club, and that individual has astigmatism. Therefore, by the definition of dominance, that makes club the dominant allele. Okay, so notice I wrote the dominant allele second. By convention, we write it first. But my point here is just simply to get you to think through the issue and not focus directly on what the convention actually says. We can sometimes avoid the convention. The other point too is, again, just because something is recognized as a large letter versus a small letter, that does not necessarily mean it's dominant. Okay, so just keep that in mind as we go. Now here's some stuff for you to practice on. I have a series of questions here. Okay, the exact same sort of situation, corneal astigmatism. Which of these is the recessive trait? All right, what about this? Suppose this guy, this gentleman named Rex, has astigmatism, but his mom did not have astigmatism. Is that sufficient information for us to determine what Rex's genotype is? Okay, that's a question. Another question. Suppose Rex has a baby brother who does not have astigmatism. Does that tell us anything about Rex's parents' genotypes? What exactly does it tell us about Rex's parents' genotypes, if anything at all? And what's the probability then that Rex's baby sister would also have astigmatism, given that we know all the rest of this information? Can you answer that question? All right, I posted those questions on the discussion, and I would like you to take a look at that, and here is the rule. The first person who posts on the discussion the correct answers to all 4 of these questions with completely and utterly correct explanations of the solutions will get 5 points of extra credit added to their first examination.